Hmm. Hi, I'll wait for a few more minutes to see if there are others joining and then I'll start.
Yes, ma'am.
All right, I think since it's 6:10, I'll get started. So, hi everyone. I'm Anakha Nair and I'm a PhD scholar at IIT Bombay. And I take these problem solving sessions for the NPTEL course, Post Harvest Operations and Processing of Fruits, Vegetables, Spices, and Plantation Crop Products. So, if you're joining for the first time, I'll just explain how these problem solving sessions work. So we have two problem solving sessions, one on Sunday and one on Tuesday. It's taken by two different TAs, but the questions are the same. So you can choose to join whichever you want depending on when you're free. And how these sessions are is that uh, regarding the module that you have studied this week, there will be some questions from that module that I'll be taking, theoretical and sometimes numerical questions also, and we'll go through them and then we'll see if you've grasped the concept or not. We will not be discussing the assignment questions, but these questions will be similar to the kind of questions that you get in the assignment. So maybe it will give you an idea of how to go about solving the assignment questions. So I'll show you the question and you can either answer by unmuting yourself, or I would prefer if you write it in the chat in case there is some network problem. So I would prefer you write the answer that you think is correct in the chat and then we can discuss what the answer is. And that is how the session will go. Please feel free to stop me anytime you have a doubt. You can unmute yourself and ask me, or you can write it in the chat. I'll try to clear that out for you. So, without further ado, let's move on to the first question for the module for the week 6, right? So, the question number 1 is, which of the following is a commonly used method for juice extraction from fruits and vegetables? And there are three options given to you: centrifugation, ultrafiltration, and hydrodiffusion press. So, take a minute to maybe think about what the answer is and just maybe write it in the chat. And just for your information, there might be many questions which either have only one answer or there can be multiple answers also. So keep that also in mind. One, all right. I'm not seeing any other answer, so maybe let's see what the correct answer is. So in this case, all three of them are correct. So you can use centrifugation, ultrafiltration, and hydrodiffusion press. All three of them you can use for juice extraction, right? So, are you familiar with centrifugation, ultrafiltration, and hydrodiffusion press, or do you want me to explain them? Are there any doubts in this question? Like, is there something you didn't understand? Sure. So, what they're asking is if you want to extract juice, what all methods can you use? So, centrifugation, as you know, it will remove the solid material from the liquid material, right? So, in that way, the juice will be extracted. In ultrafiltration, there will be a filter which will have minute pores, right? And it will only allow particles of that are below a certain size to pass through, and any other solid material will be stuck on here and they will not be allowed to pass through. So you can get juice like that, right? And in hydrodiffusion press, especially in the case of things like sugarcane, etc., you have either hot water or steam. Okay, so in a container, in a pressurized setting, you will have the fruit or vegetable of your choice, and the hydrodiffusion press will inject steam or hot water into this, and the soluble compounds will get solubilized in the water itself, and that liquid that you get, which has all these soluble compounds, you can concentrate it further to get a juice. So this the third one works for the cases where most of the molecules that you require is water soluble. In that way, you don't have to even filter it, because in this case, only things that are soluble will go into the solution, that is the water, and all the other solids will remain, and you can just remove this. Is that clear? Sure. So let's move on to the next question. Question number two is match the following type. So in question number one, you have juice extraction method, and sorry, in column number one, you have juice extraction methods, and column number two, you have the characteristics. So you have to figure out what is matching what. So maybe try figuring out what the answer is. So which juice extractor will you use for which type of uh, end product? Any idea amongst A, B, C, and D which is correct? Alright, I'm not seeing an answer, so let's maybe go and see what the answer is. So the correct answer is the option B, where the squeezer type extractor is for used for citrus fruits. So if you know how, like if you have half a lemon, how will you extract its juice? You will squeeze it using that instrument, right? Where it's like this, right? And it'll have little perforations here and the juice will flow, right? So that's a squeeze type extractor. And mostly these are used for citrus fruits because they have a lot of water content, right? Screw press, like you will see in juice shops, is usually used for continuous juice extractor. So you can keep adding fruits or vegetables and it'll keep juicing it, right? Hydraulic press is used for berries and currants. They are very small and they don't have a large amount of water content, right? Like because of their very small stature. And belt press uses pressure rollers, right? Is that clear? Do you have a doubt in this? Alright, so I'm not seeing a doubt, so I'll move on to the next question. Just stop me anytime you have a doubt, alright? So question number three is, which of the following statement is correct regarding the concentration of fruit juices? And there are three options given to you. Concentration helps in reducing the water activity of the juice. Option 2, it stabilizes the handling of the final juice product. Option number 3, concentration process can include evaporation, cryoconcentration, and membrane concentration. So out of these three, which is or are correct? There might be multiple correct answers in this. Just try guessing, or if you know the answer. Alright, 1 and 2. Yash, do you have a different answer? Same. Alright. So let's see what the answer is. So the answer is actually one, 2, 3. All three of them are correct. So concentration does help in reducing the water activity of the juice, because some amount of water will be reduced, right, when you're concentrating it. It does stabilize the handling of the final juice product, and concentration process can include evaporation to remove water, cryo concentration again to remove water, and membrane concentration to remove again water, right? So membrane concentration, if you have a semi-permeable membrane through which only water can move, right, that can also act as a concentration method. So these three of them are concentration methods for making concentrated juice, and the other two are advantages of having concentrated a juice. So it reduces the water activity of the juice and therefore increases its shelf life, and it therefore stabilizes the handling of the final juice product also, because now you have a lesser quantity to work with, right, then more. Any doubts in this? All right, I'm not seeing any doubts. So let's move on to question number 4. Question number 4 is, which of the following statements is correct about aseptic processing? Okay, and there are three options. Aseptic processing involves heating the liquid food product and cooling it before packaging. Option 2, the packaging material is sterilized before filling the product. And option number 3, aseptic packaging does not require a sterile environment. So out of these three, which is correct? Okay, someone is saying 2. 2. Okay, alright. So maybe let's see what the answer is. So 2 is an answer. You need to sterilize the packaging material also before you fill the product, and that aseptic processing involves heating the liquid food product and cooling it before packaging, like pasteurization, right? So you will heat the milk to a certain degree for a few seconds or a few minutes, depending on the heat that you're applying, and you will allow that product to cool down. And then you will pour it into packaging material which is pre sterilized. In that way, your product is also sterilized and your packaging material is also sterilized to some extent. And that will decrease the amount of spoilage that can take place and increase the shelf life. Aseptic processing does not require a sterile environment is incorrect. If it is done in a asterile or environment which is not sterile, then the chances of, even if you have sterilized your packaging material and the liquid, there are chances that there will still be microbes that go from the environment to these packaging and the liquid, and therefore the aseptic process is not complete. Right? Is there any doubt in this question? So a lot of these questions are deceptive in the way that you don't exactly know how many correct answers are there, and the questions for the assignments and for the exam if you're going to take might also be like that. So you have to make sure that you look at all the options and then see which all can fall into the correct form, right? So since I'm not seeing any doubts, I'll move on to question number 5. Question number 5 is which of the following option correctly stated which of the following options correctly stated high energy ultrasound condition for being used in ultrasound assisted extraction in fruit juices. So when you have ultrasound, what happens is it can break open the cell wall, right, of the fruit, and in that way it will release its content, and that will be the juice. So in that way you can extract juices from the fruit, right? So in this case, which of the following option is the correct energy condition that is required? And there are four options to you: 100 kilohertz, 1000 kilohertz, 20 to 500 kilohertz, 20 to 500 kilohertz, and less or more than 1 W by centimeter square. So out of these, if you have studied the module little bit more carefully, this is a theoretical question, right? So you have to remember the exact number. So this is the frequency, the kilohertz, and this is the watts per square centimeter, the W uh, by centimeter square, right? So that will give you the density or the intensity in uh, how much power is delivered per unit area. So the correct answer for this, let's see, I'm not seeing any answers in the chat. So the correct answer for this is 20 to 500 kilohertz intensity. Okay, and that is the perfect intensity for extraction of fruit juices. Now, why it is like that? There is something called low energy, right? If you remember, and this is used for reducing particle size and for collapsing any air bubbles in the liquid, right? Because a lot of fruits and vegetables, if you've noticed, once they're juiced, especially if you've noticed in Mosambি juice, etc., they will have air bubbles formed, right? But when you're packaging these juices, you will need to ensure that these air bubbles are not present, okay? Because it is taking away from the volume that of the juice that has to be present in the packaging, right? So if you use low energy, which is 100 kilohertz, right, and more than 1 W per cm square, then these are what you can do with it. Then you have high energy, which is this, and it will initiate chemical and physical changes in solid food, okay, by acoustic and hydrodynamic cavitation. So it will break the cells and release the juices, right? So the correct answer for this is high energy frequency, or 20 to 500 kilohertz. Any doubts in this question? Alright, so let's move on to the next question. Question number 6 is which of the following statement is correct regarding the quality control of fruit juices? And there are three options given to you. Total plate count should not exceed 50 colony forming units per ml. Option 2 is yeast and mold count should not exceed 2 colony forming units per ml. And 3, coliform count should be 0 in 100 ml of juice. So out of these, which is correct? Any idea what the answer is? 3, alright. So the correct answer for this is all three of them. So colony forming units is, if you have done this experiment in labs, right? You will plate a certain volume of your liquid in an agar plate, okay? And you will see after incubation colonies being formed. So each colony is right now like uh, millions of cells, thousands of cells at least, right? But each of them was initially one single cell. So it will give you an estimate of how many living organisms are present per ml, right? Because seeing one um, bacteria, especially if it is not in a concentrated sort of solution, is not easy. So that is why we do plating like this, because even if there is one bacteria, after you incubate it for multiple days, it will divide into thousands of cells, and you will be able to observe that colony. And you will know that in this case, how many colony forming units are there, right? And that means how many individual organisms were present in the 1 ml of liquid that you poured into the plate, right? So, for the quality control of fruit juices, the total plate count should not exceed 50 colony forming units per ml. Okay, that includes any kind of organism, right? Yeast and mold count should not exceed 2 colony forming units per ml. That is, any fungus initial cell should not be more than 2 colony forming units per ml, and coliform counts should be 0 in 100 ml of juice, right? And coliform is a gram negative bacteria, it's a rod shaped bacteria, right? And it is found commonly in soil, water, vegetation, and intestines of uh, mammals and other animals, right? So the presence of these coliforms will tell you that there might be possible contamination from pathogens, especially from fecal sources, right? So if you know that fertilizers are used for growing plants and vegetables and fruits, right? So there might be, even if you have cleaned them, the presence of these fecal bacteria on the fruits. So when you're juicing it, you have to ensure, especially if you're selling it, that it should be less than it should be zero. There should be no coliform uh, bacteria present in 100 ml of juice. Otherwise it indicates some form of contamination, and it's not therefore safe for drinking, right? Any doubts in this? All right, I'm not seeing any doubts, so I can move on to the next question, right? If you have any doubts, just write it in the chat, okay, so that I'll read it and I can go back and explain it in case I've missed it. So question number 7 is, which of the following statements about the problem associated with the size reduction of fruit pulp are correct? Option 1 is, too finely milled pulp can increase the percentage of suspended solids in juice. 2. Large fruit pieces in pulp lead to relatively high juice yield. Option number 3. Presence of pectinase and other degrading enzyme can be a problem with too finely milled pulp. And 4. Higher amount of suspended solids from finely milled pulp can result in bitter or undesirable taste. So out of these, which is or are true? Any idea what can be the answers or the answer? 1, 3, and 4. Alright, let's see if that is the answer. And yes, the answer is 1, 3, and 4. So if you have pulp, for example, you have orange, right, and you're making juice of it, and you have pulp. So if you finely mill it, if you break it into many different pieces for palatability, so that it's easier for you to drink, right, it can increase the percentage of suspended solid in the liquid or in the juice. So since this one piece has become multiple pieces, right? So it will increase the percentage of solids in the juice. Large fruit pieces in pulp lead to relatively high juice yield is incorrect. It decreases the juice yield, right? Because this fruit piece has not been converted or extracted properly into a juice. Option number three, presence of pectinase and other degrading enzyme can be a problem with too finely milled pulp. So these will be containing cells, right? If you finely mill them, these cells will break open and they can release different kinds of enzymes like pectinase and other enzymes which can degrade other valuable compounds in the juice. And option 4, high amount of suspended solids from finely milled pulp can result in bitter or desirable taste, right? Especially in the case of citrus fruits, imagine you have the peel of it, some amount of peel, and you know they can be bitter, right? And the fact that you have suspended them very, like, the process has included pulp in very small pieces that you can't even remove it, will ensure that you will, when you're drinking it, you will get the bitter taste or the undesirable taste of these extra ingredients also, right? So any doubt in this question? All right, I'm not seeing any doubts, so maybe I'll move on. So let's move on to question number 8. So question number 8 is, which of the following statements about pan or kettle evaporators are correct? There are four options given to you. Pan or kettle evaporators use a hemispherical shell, that is this shape, right, made of stainless steel. Option 2, they are suitable for concentration of thermally labile products. Option 3, heating in pan evaporation is done by steam circulating in coils or by copper heating coils. And pan evaporators are not suitable for concentration of juices, jams, sauces, and confection products. So, what do you think the correct answer or the correct answers is? So, this is a pan or kettle evaporator, right? So you have a mixture, an aqueous extract of the juice that you're trying to concentrate, and the vapors of the solvent will evaporate, and you will be left with a concentrated form of what you originally started off with, right? So you have a steam inlet which will heat this material inside slowly. You have a outlet for non-condensed gases. You have outlet for condensates. You have outlet for product. So after it has gone down in amount, right, you can open this valve and release the product, right? So I'm not seeing any answers, so let's maybe see what the correct answers are. So pan and kettle evaporators use a hemispherical shell made of stainless steel, like you can see here, that is true, right? They are suitable for concentration of thermally labile products, which is not correct. So thermally labile products are those which get degraded when they are when they come in contact with heat, right? So this can be used only for thermally non-labile products. That means they will not get degraded when they are heated, right? Option 3 is correct, which is that heating in pan evaporator is done by steam circulating in coils, like in this case, or a copper coil. So you can have a copper coil below the steel surface of the vessel here, and that will also work in the same way, right? And lastly, pan evaporators are not suitable for concentration of juices, sauces, jams, and confection products is also incorrect. It is suitable for concentration of juices, sauces, jams, and confection products. Does anyone have any doubt in this question? All right, I'm not seeing anything. Let's move on to question number 9. So, question number 9, oh, labile. If that is a doubt, you want to know what labile means in this case? Okay, labile in this case means thermally labile means compounds that will get degraded. Okay, that means they are susceptible to degradation because of heat. The compound is not of a very stable structure, so they will get degraded when you degraded, or there will be some change chemically when you apply heat to them. There are very stable compounds, thermal stable compounds, and there are thermal labile compounds. So labile compounds are those which get degraded when you apply heat to them, and thermally stable compounds are those which don't get degraded when you apply heat to them. And this is a normal threshold of temperature, right? So a lot of compounds, if you reach very very high, obviously temperature, they can get degraded, right? But within the range of heating that we're talking about, in that we can do in kitchens or in cases like this, in within that range they should be either thermally stable or thermally labile, right? Is that clear? Labile by itself means unstable or easily altered, right? So this the labile portion means unstable or easily altered, right? And the thermally in front of it means that the instability or the alteration is because of application of heat, right? Yeah. So let's move on to the next question. Question number 9 is, which of the following statements are true regarding juice clarification? So when you make juice, it might be murky in appearance. That means it is not clear, right? It can appear murky or cloudy in appearance, right? And you do clarification, which utensils can be used for labile products. So in that case, it's not a utensil, it is just you can't use heat, right? So you can't use concentration. Yeah. So in this case, it just means that you can't use heat as a way to concentrate. So in this way, you are removing water vapors using heat, right? So you can use something like concentration. So instead of heating, you are freezing the solution, and the water uh, will become ice solids, and everything else will get separated. So in that way, you're not heating the liquid, right? So it can be used for thermally labile products. Just heat cannot be used. Any other doubt? All right. So let's move on to the next question. This was regarding clarification. Which of the following statements are true regarding juice clarification? So there are three options. Clarification improves the visual appeal of the juice. Option number 2, it increases the shelf life by removing suspended particles. And option number 3, clarified juice has a higher nutritional value compared to unclarified juice. So what do you think the correct answer is, or the correct answers are? 1 and 2? Yes. So the correct answer is in fact 1 and 2. So clarification, like I was saying, will improve the visual appeal of the juice. So instead of appearing cloudy or murky, it will appear as a clear liquid, and that is much more desirable for consumers, right? And it will increase the shelf life by removing suspended particles that can result in spoilage, right? So that is also true. Clarified juice does not have a higher nutritional value compared to unclarified juice. You are removing things from it, you're not adding things. So it cannot have a higher nutritional value compared to unclarified juice. So this is not correct. Alright. Any doubts in this? Alright. So let's move on to question number 10. Question number 10 is, which of the following statements about cryo concentration, also known as freeze concentration, are correct? There are four options given to you. Cryoconcentration involves the removal of pure water in the form of ice crystals at sub-zero temperatures. Option number two, cryoconcentration is particularly suitable for the concentration of thermosensitive biological compounds. Option three, cryoconcentration harms the integrity of biological material. And option number four, cryoconcentration uses components like crystallizers and washing columns. So out of this, which is uh, correct and which is incorrect? 4. All right. So let's see how a cryo concentrator looks like. So you have fresh juice, right? And you have crystal formation at 4 degrees Celsius, right? So crystals of water, maybe 1, 2, sure. Let's see. Crystals are formed. Then you remove the ice crystals and you wash it, right? And you get the concentrated product, and you can remove the ice, right? So while the ice is forming, there might be some solids that get stuck in it, right? So you will use a wash column to remove anything stuck to the ice so that you get pure ice here. Okay, in that way you're removing water content from the uh, juice, but you are keeping all the other kinds of compounds. Okay, so you are correct. 1, 2 and 4 is correct. So cryo concentration does use components like crystallizers and washing columns. Yes, cryo concentration involves the removal of pure water in the form of ice crystals at sub-zero temperature. That is also correct, because you have to ensure only ice, it is at a temperature where only water is converted into ice crystals. If it is very low, for example liquid nitrogen and all, there might be other compounds that also get frozen, right? So you just want to remove water and nothing else, so that therefore you will use the threshold where water freezes, right? And cryo concentration is, yeah, cryoconcentration is particularly suitable for concentration of thermosensitive biological compounds. So, like we were saying, in the kettle evaporation method, we can't do it for thermolabile or thermosensitive compounds. So, in that case, you can use this method. Since it is cooling, they are not affected by cold temperatures. They're only affected by warm or hot temperatures, right? So 1, 2, and 4 is correct. 3 is incorrect. Cryo concentration does not harm the integrity of the biological material in any way. It is just freezing the water. Right? Any doubts in this? All right. So I'm assuming no doubts. Let's move on to question number 11. Question number 11 is, given the following values for an indirect heat exchanger, the mass flow rate of hot liquid mH is 2 kg per second. The specific heat of hot liquid is CPH, 4 kg per 4 kilojoules per kg degree Celsius. The inlet temperature of hot liquid (THI) is 80 degrees Celsius and exit temperature of hot liquid is 60 degrees Celsius. You have to calculate the heat transfer rate. Okay. So maybe let's go directly to the answer. So what they're saying is, in indirect heat exchanger, where imagine this is your material and you have water flowing, but there is something between them, between the hot water flowing and your material. So the water is not directly in contact, or the heating element is not directly in contact with your material. So there is only transfer of heat. So it is an indirect heat exchanger, right? There is no direct contact between the heating element and your material. So heat is being transferred, for example, in this case, from water through whatever barrier was present here, right? So in this case, the mass flow rate of hot liquid is 2 kg per second. Specific heat of hot liquid is 4 kilojoule per kg per degree Celsius. Uh, Inlet temperature is 80 degrees Celsius and exit temperature is 60 degrees Celsius. So if you know the formula, you can calculate this. So the heat transfer rate is given by this formula, right? And you have to substitute the given values. So mH, that is the mass flow rate of the hot liquid, which was 2 kg per second, into Cph, which is the specific heat, right? So this is the flow rate, mass flow rate. This is the specific heat, right? The inlet temperature and the outlet temperature, right? So if you input the values in this case, you will get an answer of 160 kilojoule per second. Since 1 kilojoule is equal to 1 kilowatt, watt, and they were asking for the heat transfer rate and the answers are given in kilowatts, you will convert it and you will get the same answer, which is 160 kilowatt instead of kilojoules per second. Right? So that is the correct answer. Is there any doubt in this question? Yes, I see that Yash has gotten the answer. That is great. Any doubts? Naveen, do you have any doubt in this question? Alright, so I'm not seeing any doubts as such. So maybe let's move on to the next question. So question number 12 is, which of the following statements about heat sterilization of aseptic packaging material and equipment is correct? Option 1 is, product supply lines and filters are commonly sterilized by moist heat in the form of hot water or saturated steam under pressure. Okay. Option 2, dry heat in the form of superheated steam or hot air may also be used to sterilize equipment. Option 3 is, systems employing moist heat are sterilized at temperatures ranging from 150 to 160 degrees Celsius. And option 4 is, sterilization of air by incineration usually is conducted at temperatures ranging from 260 to 315 degrees Celsius. So out of these, can you guess which are correct? Alright, I'm not seeing an answer. So maybe let's move on to see what the answer is. So for sterilization of aseptic packaging material and equipment, Product supply lines and fillers are commonly sterilized by moist heat. Okay, in the form of hot water or yeah, network issue. Yeah, could be my problem. No worries. Uh, so you're able to see now, right? And hear now, right? Are you able to see the PPT and hear what I'm saying? Am I not audible? I'm not seeing anything in the chat. Am I audible now? Are you able to see the screen? That is the PPT. Oh no. Yeah, I think there is something wrong with my net. That is why there is the network issue. All right. So I'm hoping that you are able to see the PPT and hear my voice now. So, like I was saying, the product supply lines and the fillers are commonly sterilized by moist heat in the form of hot water or steam under pressure. Okay, since they are not a part of the packaging technically, they are just part of the supply line, and it is uh, you know the amount of water is not a problem there. So even if there is some small amount of remaining water in on top of these instruments, it is not a problem. You can't do the similar thing with the actual product because it will increase the water content of the product. So for product supply lines and fillers, sterilization by moist heat as hot water or steam under pressure is used. That is correct. Dry heat in the form of superheated steam or hot air may be used to sterilize equipment, especially electrical equipments that cannot be sterilized using moist heat. Right? If there is water present, it will get short circuited. Right? So that is why dry heat will be used in these cases. System employing moist heat are sterilized at temperatures ranging from 150 to 160 degrees Celsius is not correct, since we are talking about steam, and we are talking about steam is mostly at the temperature of 100 degrees Celsius, right? And with pressure, the maximum temperature that you can reach, especially if you've ever autoclaved anything, you will know that it is 121. We autoclave at 121 degrees Celsius, right? So the range is 121 to 129 degrees Celsius for moist heat sterilization. Okay, not 150 to 160 degrees Celsius. So this is incorrect. And option 4, sterilization of air by incineration is usually conducted at temperatures ranging from 260 to 315 degrees Celsius. All right. Is there any doubt in this question? All right. So I'm not seeing any doubt. I'll move on to question number 13 then. So question number 13 is, which of the following statements are true related to the use of radiation in food processing? So option 1 is, due to the penetration power of gamma radiation, packages are treated in bulk at commercial irradiators. Option 2, a dose of approximately 1.5 millirad is commonly used to decontaminate containers for acid and acidified foods. Option 3, doses required to sterilize containers for use with low acid food are considerably higher than those required for acid and acidified food. So out of these, which is or are correct? Do you have any guesses or on what the answer could be? One, all right. So let's see what the answer is in this case. So all three of them are correct. So gamma irradiation can pass through surfaces, right? So even if you have a packaging on top of your food, it can pass through that and directly sterilize the surface and the interior of the fruits, right? So therefore, because of this, you can do something that packages are treated in bulk at commercial irradiators. So you can have packages on top of each other, and in other cases like UV, etc., this will not work because it only works on the surface level. But since gamma radiation can pass through these surfaces, you can sterilize the entire containment or shipment, right? So the first one is correct. Second is also correct. An approximate dose of 1.5 millirads is commonly used to decontaminate containers that will be used for acid or acidified food. Now, acid and acidified food is less prone to spoilage. That is why it requires less amount of gamma irradiation for decontamination. But similarly, the doses required to sterilize containers that will be used for low acid food, which are more easily spoiled, is higher. You will use higher amount of gamma radiation to sterilize than in the case where the food that you were packaging was acid or acidified food. All right. So all three of them are correct. Any doubts in this question? All right. I don't think I'm seeing anything, so I'll move on to question number 14. Question number 14 is, which of the following statements about nectar is correct? So there are four options. Nectar is made from fruits with high acidity or fruit pulp by adding water and sugar. Option 2, nectar is clearer and smoother than fresh fruit juice and doesn't need to be diluted. Option 3, nectar should contain at least 10% fruit or fruit juice or pulp and 5% total solids, soluble solids, right? And option 4, nectar contains only fruit juice without any additives. So out of these four, which is or are correct? 1, 2, 3, 4. All right, let's see what the answer is. So the correct answer in this case is nectar is made from fruits with high acidity or pulp by adding water and sugar. So if you have fruit juices which are high in acidity and fruit pulp, so there are certain fruit juices that you cannot have without diluting it, right? So this is the case for those which have high acidity and fruit pulp, and you add water, and to increase the taste you will add sugar also. So that is nectar. So for example, peach nectar, right? So peach itself doesn't have a lot of water content as such. So a juice of peach is uh, for better than that. You will have uh, uh, you know fruit juice or the crushed fruit, and you will add water and sugar to it to get the nectar, right? Nectar is clearer and smoother than fresh juice and doesn't need to be diluted. So nectar is actually thicker than fruit juice, and needs to be diluted down. Okay, so this is incorrect. It should contain at least 20% fruit and at least 15% total soluble solids. Only then is it considered nectar. Since the numbers are incorrect in this, option C is also not correct. And option D obviously is incorrect. Nectar contains only fruit juice without any additives is not correct. Nectar contains fruit pulp, fruit fiber, pectin, sugar, preservatives, etc. So the only correct option in this case was option A. Any doubts in this question? All right, I'll move on to the next question. So let's move on to question number 15. What does RTS stand for in the context of beverages? There are four options given to you: ready to serve, ready to store, ready to season, ready to sip. So this is a relatively simple question. You can guess the answer, I think, easily. So what do you think the correct answer is? Right. So the correct answer is A, ready to serve. So in context of beverages, it is where you don't have to do any additional steps before serving it to someone. So that is RTS, ready to serve. Then there might be beverages which you have to do some modification. You might need to add sugar, you might need to add water, etc. And those are not a part of this ready to serve, right? So ready to serve is the context in this case. It is not ready to store or ready to season or ready to sip, right? So let's move on to the next question. I'm hoping there are no doubts in this, right? So question number 16 is again a match the following. You have to match the preservatives commonly used in beverages, which is given in column 1, with their function given in column 2. So what do you think the correct answer is? Try matching one or two which you are very sure about, and then from the options given, see which of these match the two that you know of. It will make guessing the rest, in case you don't know the answer, much more easier. Right, I'm not seeing an answer, so let's see maybe what the answer is. So answer is option A in this case, and the thing you should have focused on was ascorbic acid and citric acid, right? So option number 3, ascorbic acid, we know is a antioxidant. It's a common antioxidant, right? And citric acid will provide acidity and preserve the color, right? Now the other two that that you needed to see is potassium sorbate and sodium benzoate. So if you didn't know the other two, you could have guessed from the ascorbic acid and the citric acid what the correct answer is, because this form was not in any other option, right? So sodium benzoate will act as a microbial growth inhibitor, and potassium sorbate will prevent mold and yeast from growing. Any doubts in this question? Alright, let's move on to question number 17. So question number 17 is, which of the following statements about titratable acidity is correct? Right? So if you remember from your chemistry class, you would do titration, right? To figure out the acidic content or the concentration of certain liquids, or the basic concentration of liquids, etc., right? So, which of the following statements about titratable acidity is correct? There are four options given to you. Titratable acidity measures the concentration of hydrogen ion in a food sample. Option 2 is it is an important parameter in determining fruit maturity and sour taste in citrus fruits. Option 3, juices with low titratable acidity are difficult for saliva to neutralize. And option 4, immature fruits will normally have a high sugar to acid ratio. So out of these, which do you think is or are correct? All right. So I'm not seeing anything. I'll Let's see what the answer is. 2 and 3. All right. So the answer for this is option B. So titratable acidity measures the concentration of hydrogen ions is not correct. In this case, it is not the hydrogen ions that is being sampled. So hydrogen ions does give the pH of a solution, free hydrogen ions, but that is not the total acid content of a liquid, right? So that is measured in TTA. So titratable acidity measures the amount of acid or acids present, and this can be acidic salts, etc., which are not forming hydrogen ions as such, right? In a food sample, it doesn't necessarily need to be the concentration of hydrogen ions in a food sample. So this is not correct. It is an important parameter in determining fruit maturity and sour citrus taste in citrus fruits, because citrus fruits are more acidic in nature, right? And fruits which are over ripened are also more acidic in nature because the sugars would have been converted to acids, right, by the enzymes. So the more ripe the fruit is, the more acidic it will be. Juices with low titratable acidity are difficult for saliva to neutralize is not correct. So juices which will have high titratability acid rate, that means they have high acid content, are difficult for saliva to neutralize and therefore will potentially damage your teeth. The acid content of these juices will damage your teeth. And immature fruits will normally have uh, low sugar to acid ratio, right? Pure fruit juices have high titratable acidity, and immature fruits will normally have low sugar to acid ratio instead of high sugar to acid ratio. Mature fruits will have a high sugar to acid ratio, but immature fruits will have a low sugar to acid ratio. Alright. So let's move on to the next question if there are no doubts in this. Question number 18 is, which of the following statements about the safety of fruit juices is true? Option 1, pasteurization destroys pathogen. Option 2, fruit juices do not require any form of preservation. And option 3, proper packaging prevents contamination. So, out of this, which is or are correct? 1 and 3, yes. So, the correct answer is pasteurization does destroy pathogen. Proper packaging does prevent contamination. But option 2 is incorrect, that is, fruit juices do not require any form of preservation, is incorrect. So let's move on to question number 19 then. Question number 19 is, which of the following statements about the effect of evaporative concentration process on quality attributes is correct? So evaporative concentration was the process that we've seen, right? In which you have a slurry and you will evaporate it so that it becomes more concentrated, right? Without volatile recovery, loss of aroma compounds include ester, aldehydes, terpenes, reduce the flavor of concentrated juices. Option 2, darkening of the color of foods occurs partially because of decrease in the concentration of solids. Option 3, reduction in water activity promotes chemical changes such as Maillard's browning, causing changes in anthocyanins and other pigment in fruit products. And option 4, improperly selected concentration methods may be beneficial to the product's sensory and nutritional profile. This might be a slightly difficult question to understand. Maybe try reading through it once more by yourself from the screen and then try to guess what the answer is. Alright, I'm not seeing any answers, so maybe let's go and see. So the flavor of the juice, etc., depends on the compounds, right? And many of those compounds can be, like we were talking, thermal labile, right? So what happens is when you heat them, they will get evaporated. And since these compounds have gotten evaporated, it might change the flavor profile of the juice that you were concentrating, right? So what can be done is you can recover these volatile compounds, that is you can collect the vapors, and from the water you can separate these volatile compounds and re-add it to the juice if needed, right? So without volatile recovery, loss of aroma compounds such as esters, aldehyde and terpenes reduce the flavor of the concentrated juices. That is correct. So once you start concentrating something, you will notice that its color becomes darker, right? So that darkening of color occurs partially because of decrease in concentration of solids is incorrect. It happens because of an increase in the concentration of solids when compared to the liquids in that concentrate, right? So that is incorrect. Option 3, that is reduction in water activity, promotes chemical changes such as Maillard's browning and causes changes to anthocyanin and other pigments in fruit products, which is also correct. So a lot of time when you heat fruit, they don't uh, continue to remain the color that they were originally, right? Because the color compounds in this case due to heat will change, especially due to concentration, I mean reactions like Maillard's browning, etc., right? So a lot of the time when you're cooking food, you will notice the browning that happens, right? That is the Maillard browning, right? So this is correct. Option 4, which is improperly selected concentration methods may be beneficial to the product sensory and nutritional profile, is incorrect. So you have to carefully select the concentration method, right? Depending on what the flavor profile is, what the compounds that give the flavor is, because otherwise you might lose compounds. If, for example, if you were using heating concentration by heating the material, you might lose compounds that are important for its taste, right? And flavor. So that's why it may be detrimental to the product sensory and nutritional profile, right? Is there any doubt in this question? So this is also, option 4 is also incorrect. So I'm not seeing any doubts as such for now. So let's move on to the final question for today, which is which of the following statements about the packaging of fruit juices is correct? And there are three options. Packaging material should be impermeable to gases. Option 2 is packaging should protect the juice from light. And option 3 is packaging material do not affect the shelf life of the juice. So out of these, which are correct? 1 and 2. Yes. So the correct answer is packaging material should be impermeable to gases. If it is permeable to gases from the atmosphere, what happens is oxidation or spoilage of the juices can happen, right? So it has to be impermeable to the gases. So gas exchange within the container and outside the environment cannot take place. And packaging should protect the juice from light, which is also correct because light a lot of times can degrade color and nutrients, right? So it has to be. So if you see a lot of packages behind them, it will be written keep away from direct sunlight, and that is because of this, right? And option number three, which is packaging material do not affect shelf life of fruit juices, is incorrect. It does affect the shelf life of fruit juices. Any doubts in this question? This was the last question for today. Is there any doubt in any of the questions or any doubt that you want me to clarify? If not, then that is the end of the session for today. You can leave the meeting if you don't have any doubts. Thank you so much for joining. Thank you. If there are any doubts, you can ask me. Otherwise, you can leave the meeting. Thank you so much. For what reason should I turn on the camera? I think I'm slightly uncomfortable with turning on the camera. That's why I don't do that. And my network is also not great. So I'm going to say no to that. Any other doubts? Do you have any doubts, Yash? If not, you can leave the meeting. Yes. Hi, the session for today is over. I finished all the 20 questions. Do you have any other doubts? Because I'll be uploading this online. You can watch the session from YouTube. Do you have any doubts apart from this? All right. So I'll upload this and the link will be shared with you. You can go and watch the entire session on YouTube whenever you're free. All right.